off throughout the session so that we can listen to the speaker without any distractions or background noise. And thank you so much for joining us firstly, and our apologies that we could not conduct this in the regular format on the Zoom webinar series call. But essentially what we're going to do today is in the first 5 minutes, I'll give a quick introduction about the session. So at the Good Food Institute India, like a lot of members who've joined us today know and who've been attending for the past 20 webinars also know, we've been essentially building the smart protein sector for India. So that means that we are focusing on plant-based meat, eggs, and dairy, cultivated meat, eggs, and dairy, and fermentation-derived meat, eggs, and dairy products. And that's essentially what we, kind of the three key modalities of production of alternative proteins or smart protein sector, as we call it in India. Having said that, today we are here for GFI India's Technological Review of Fungi, where we have studied the fungi supply chain and many other aspects of it, value chain of it, to kind of understand its specific applications to the smart protein sector. And our guest speaker for today is essentially a GFI India team member, Rama Devi Tentu, who's our GFI India SciTech Research Fellow and who's led this research and has in the past few months extensively analyzed this kind of a value chain and its application in the alternative protein domain. So Rama, do you want to share your screen and get started with the presentation?
Thank you, Shardul. So yeah, let me just share my screen. Shardul, can you confirm if my screen is visible? Shardul, can you confirm if my screen is visible?
It's visible.
Yes. Hi. Yeah. Okay. So thank you everyone for joining us, and apologies again for the non-functional Zoom link, but we are glad to have you here. As you would already know, today's webinar is basically on exploring different functionalities and applications of fungi in the smart protein sector and how these can be leveraged by especially the Indian entrepreneurs, researchers, etc., who want to venture into this space. So the way today's webinar is structured is that I'll give you a very brief idea in terms of how the smart protein landscape looks like and how does fungi fit into it, what do we mean by this microorganism known fungi, and subsequently we are going to talk about the existing applications of fungi. We look at the value chain right from strain selection to product development, and then we are going to specifically talk about this applications of fungi in the smart protein sector. Based on the analysis and study that we have conducted over the last few months, we'll be presenting some of the opportunities across the value chain, both which are applicable globally as well as locally in India. And last but not the least, we'll also be launching the strategic analysis of fungi, which basically this presentation is a summary of the report. And for any in-depth insights, you'll be able to access the report after this session. So let's dive right into it. As Shardul just mentioned, and many of you who are working in the alternative protein sector or are interested in the smart protein sector already know, there are three technological routes that you can take to create these smart protein products. One is fermentation, which is the focus of the webinar today, wherein you're looking at microorganisms to either create ingredients which can be used to make alternative meat, egg, dairy products, or improve the quality of existing ingredients like plant-based proteins. Then the second set of ingredients is second set of technology is plant-based proteins, which is using the plant-based proteins to and transforming them in such a way that they can mimic animal-based proteins. Last but not the least is cultivated meat, which is basically growing an animal tissue outside an animal in a bioreactor. So being able to do tissue engineering in such a way that you can create animal muscle tissue out as a separate process itself. And I would like to remind all of you about GFI's theory of change, which is central to the smart protein sector, which is make things that people would want. So we don't want people to start eating chickpeas instead of chicken, but we would rather make a chicken product, a chicken which is made out of chickpeas. So keeping that in mind, going forward, we need to rely on various sources of proteins. And as many of you would know who are working in this sector, the most commonly used protein sources right now are mostly plant-based and mostly limited to legumes and pulses, and especially soy and pea. There are other sources like in cereals you have rice, wheat, maize, which have relatively less amount of proteins but can show different functionalities. We have pseudo cereals as well as nuts and seeds. While plant-based protein sources are still well known, there are newer sources of proteins which are emerging and giving uh, uh, good results both at a research level as well as in terms of applications by companies who have tried to establish these platforms. So there's microalgae, seaweed, fungi, which is the focus of today's webinar, and animal cell culture, which is nothing but cultivated meat applications. So looking at different protein sources is extremely critical for the sector to be able to leverage different functionalities to meet different culinary preferences that people have. And given in India, the diversity of food that we eat and the diversity of taste that is expected, we need to look at different sources. And with this, I would get started on explaining you about what this microorganism is all about, because that's essential to be able to understand how do we leverage the properties of this organism for creating the kind of food that we want. Um, so this is this figure is might remind you of the story about an elephant which is being touched by many blind men who are trying to figure out what an elephant is. And depending on which part of the elephant they touch, they have different opinions in terms of what it really is. Many of you, if I were to today right now ask you what fungi is, for most of you, the first thing that will come in your mind is a bread with a mold, and that's what we are taught when we are kids studying biology. So the first thing comes to your mind is it's a pathogen, but then it's much more than that. And I would explain some of these things in a bit, but fungi has applications in medicine. It has been traditionally used to make antibiotics and pharmaceutical products. It makes food better. So there is no better example than yeast being used for baking applications. It's obviously we are having this discussion because it has applications in the smart protein sector. It also has some weird applications, like you can see this dead ant, which has been hijacked by a filamentous fungi. So it could be a killing machine depending on what species you are looking at. So in all, this is a work of art, and fungi is like extremely versatile organism which has various applications. Coming to what this organism is really like how does it look like, what is it composition, how does it grow? Fungi are basically heterotrophic organisms, which means that they unlike plants, they don't depend on sunlight. Rather, they depend on organic carbon sources for growth. They are eukaryotic in nature, meaning that their nucleus has a cell membrane. They are both unicellular and multicellular in nature, which can be surprising to some people because either we think of fungi as yeast or as mushroom, but this particular microorganisms has species which can be unicellular or multicellular, and sometimes can change between one kind of an organism to another depending on the conditions. In terms of the way fungi grows, it relies on food that a typical organism would ingest the food first and then digest it using the enzymes inside its body. But in case of fungi, as many of you would know, it like especially mushrooms, which rely on dead, decayed matter, they first digest it by releasing enzymes outside their body, and then they ingest the simpler compounds that are formed by the degradation caused by the enzymes. And last but not the least, the morphology meaning that the forms in which fungi can manifest itself are quite varied. And here I would like to classify them into three parts just for simplicity's sake. So one is unicellular fungi, which is and a classical example of that is yeast. So on the left hand side, the top diagram that you can see, there's a unicellular fungi, meaning that it has only one cell, and a classical example being yeast. The second one is filamentous fungi, and as the name suggests, filamentous means that it has this structure which is long and consists of filaments, and then there could be a collection of these filaments which could be clubbed together and called mycelia. And the last but not the least is fruiting bodies. Fruiting bodies can be equated to mushrooms to some extent, and these mushrooms, although they look like, they are a separate organism altogether, but they really are a collection of filamentous fungi, the bundles of filamentous fungi or mycelia. So the diagram below also shows like how, a reproductive cycle for mushroom looks like. So from spores, you have filamentous fungi coming out, you have hyphae, mycelia, which all bundle up together and make this fruiting body. So filamentous fungi is not very different from the fruiting bodies that we see in the mushroom. It's just a particular structure that it has formed. Now, having explained to you some of these things and also shown you the diagram of the elephant, can you think of some of the examples of fungi? Ideally, in Zoom, I would have loved to see the chat, but I would move on, assuming that you already know some of these applications. But I'm sure many of you would be surprised to know how frequently fungi has been used in our food and how much it has been used for such a long time. Many of the questions that we received for this webinar included, is it safe to consume fungi? Would it cause diseases if we eat this? And when you look at the list of food in which this has actually gone into, you would be surprised that we have already been consuming this for a very long time. So there is cheese. So there are different types of cheese like Roquefort cheese or Camembert cheese, which are made using filamentous fungi. Beer, wine, spirits are all brewed using brewer's yeast. Bread, of course. Then tempeh, soy sauce, tofu, all rely on fermentation as a platform, and tempeh specifically relies on growth of filamentous fungi. Enzymes and food processing aids are again obtained from fungi, and this is again like the excretion of the enzymes for the digestion of the food that it needs, and it's extracted for food application purposes. Fungi can also be used to produce vitamins, natural flavors, as well as pigments. And a classical example of a new kind of food that has been consumed is corn in the UK, where the fungal biomass is used to make alternative meat analogues like meatballs, mince burger patties, etc. And this has been since the 1985. Having said this, it's not just food to which fungi has applications. As I mentioned earlier, it's been used for many years for production of antibiotics. Corn is where it is used as a meat analog already. We have enzymes which have found applications across pharmaceutical industry, food, as well as home care applications like in detergents. More recently, we have seen that there are furniture and textiles, and even buildings for that matter, which can be made out of fungi. Having said this, let's focus on the smart protein sector and get into the basics of what does it take to grow this organism. Like what does it take for me to create a product out of fungi? And the first step here is to understand that fermentation is the basic process for production of any microorganism. So fermentation means production of microbial species, which could either be consumed as is or be used to extract a particular component out of it. While fermentation as a technology has existed for many years, the applications in the smart protein sector are fairly recent. And despite being fairly recent, there have been quite some advancements, and I'll touch upon it in a bit. If you look at fermentation, there are three main ways in which you can utilize fermentation as a platform. And first would be traditional fermentation. So when I mentioned about products like tempeh, soy sauce, or even tofu, for example, alcoholic beverages and cheese, this involves traditional fermentation. So the idea is that you use the microorganism to transform a given substrate. So the application is not to consume the microorganism, but the application is to improve the quality of the substrate uh, uh, on which the microorganism grows. Second is biomass fermentation, which basically the end application is to be able to utilize the microbial biomass for consumption purposes. And this is what Quant does and many other companies, which I will touch upon later. And these can also be used to specifically have produce high protein content organisms and then extract from it. Then there is precision fermentation, which is which basically involves genetic engineering. So you play around with the genetic makeup of the microorganisms in such a way that it expresses a compound of interest. So it could be growth factors for cultivated meat applications, enzymes, flavoring agents, pigments, amino acids, vitamins, anything that you can produce from it. You can produce egg white proteins, for example, from using precision fermentation. And this is not something I'm talking which is futuristic or something. This is already here. People are already making products using these techniques. Um, so on the screen, you see some of these companies which are using traditional fermentation as a platform, as a technology. And I would like to highlight Mycotechnology specifically here. They are using shiitake mushrooms uh, to uh, to improve the quality of rice and pea protein, which then go into creating plant-based meat products. And the whole idea is that the fermentation using shiitake mushrooms in sub liquid state fermentation improve the organoliptic properties and improve the digestibility and nutritional profile of the ingredients. So this application is already being explored. The second application, which is biomass fermentation, is something which is catching up to speed at a very fast pace right now. In the 1970s, Quorn was the company which started with this ambitious project to cultivate filamentous fungi to meet the nutritional needs of people. And now we have companies like Meaty Foods, Mycorrhiza, and many others who are trying to use filamentous fungi whole biomass to create meat analogs specifically. We also have companies like Nature's Find and Atlas Food, which are using not liquid state fermentation but solid state fermentation in a way that you can use the fruiting bodies which have similar taste profile to that as that of a meat, and utilizing the inherent properties of mycelium biomass which comes from mushroom to directly for direct applications in the smart protein sector. In terms of precision fermentation, we have companies like Clara Foods, which creates egg white proteins from which obtains egg white proteins from yeast. We have Perfect Day, which is using filamentous fungi for producing milk proteins. And last, we have Richcore Life Sciences, which is an Indian-based company, and they are producing growth factors which go into cultivated meat applications, and again, they're using filamentous fungi. I just want to mention that there are regulatory constraints around using genetically modified organisms in India. In general, the perception is not so great, and hence, like, there are not many companies in India which are specifically looking at precision fermentation as a technology. Having said that, there are ways to screen existing natural microorganisms or fungal strains which can produce some of these compounds, and that could be one of the routes to explore for the Indian market. Having said this, these companies are already established. There has been some major activity in this space which just says that how important fungi as a microbial platform is becoming for fermentation as a route. So we recently had the announcement where Unilever has partnered with Enough, formerly known as 3F Bio. Enough is again using liquid state fermentation of filamentous fungi and doing it in a much more sustainable way. And this means that they will be able to scale up their operations in the coming future. We have Mycorrhiza, which again is using filamentous fungi, and they are trying to use side streams and novel substrates for production of filamentous fungi, and they have raised about $9 million recently as pre-seed funding. We have Nature's Find, which uses an extremophile strain of fungus, and this they have extracted from the springs of the Yellowstone National Park and have received no questions letter from FDA, which is almost like a green light in response to their generally recognized as safe application. We have fuming ingredients, and this announcement is a bit dated. This is dated to last year, and fuming ingredients raised €500,000 in investment from AB and Bev, which obviously has expertise in fermentation, and they've recognized the potential of this technology as well. Now coming to like what exactly would it look like when you are trying to make products out of fungi. So we'll take a value chain approach by looking at each of the steps involved in starting from strain selection to building an end product. So the key steps, there are three key steps that are involved in production. So first is strain selection. This is the most important step, because what this step determines is everything that happens in the downstream. So be it in terms of determining what would be the efficiency of the process, what would be the quality of protein that is produced, what would be the nutritional value, the toxicity profile, allergen profile, as well as the downstream extraction processes will be defined by the kind of strain you use. So selecting the strain is the most critical part of the value chain. Once you've selected the strain, the next step is obviously production, choosing the right production system for fermentation. So there are two routes that you can take. One is liquid state or semi-solid state fermentation, which is typically used for production of yeast and filamentous fungi. And second is solid state fermentation, which is typically used for mushroom cultivation. There is one component which kind of links, which kind of overlaps between strain selection as well as production, is that of feedstock optimization, which is extremely critical to ensure that you are getting the right yield, efficiency, and quality of protein. Last but not the least, the downstream operations also are quite energy intensive, depending on the application area that you are targeting and species that you're looking at. So typically for liquid state fermentation, harvesting is an important step, because you have to concentrate the solid biomass in the liquid broth. So you have to separate the solids from the liquids and then eventually concentrate it. You also have to make sure that the RNA content, which is typically high in case of yeast and filamentous fungi due to high growth rates, needs to be reduced for human consumption. This is a very critical step, given that there are regulations around how much RNA content could be there, and it could be actually harmful for consumption if it is above 2% in terms of daily intake. Then in downstream operations, if you are looking at extraction of specific compounds like proteins, enzymes, and amino acids, hydrosylates, etc., you need to follow a typical extraction process in order to be able to utilize these functional ingredients. Now talking about each of these specific steps. So in terms of strain selection, if you have to screen for different efficient strains, you need to look at different parameters and optimize for these. So the way when you screen the kind of species that you need to utilize, you need to look at the growth rate, that the growth rate is high, so that you have high productivity rates. The protein content is high. There is history of safe use, which means that there is evidence that this has been used previously in food products and hence will be safe for use in the future. In case you are looking at new strains, you need to ensure that it doesn't have mycotoxins, it doesn't have allergenicity potential, and you need to make sure that it is safe for consumption and prove the same, which is going to be a lengthy process from a regulatory perspective. You also need to look at substrate efficiency, which is linked to feedstock consumption. So you need to make sure that for the particular feedstock that you have, in case you have constraints on it, it should be able to utilize it in an effective fashion. Absence of toxins, it's a no-brainer. You need to ensure that there are no mycotoxins. And last but not the least, you need to look at how easy it is to perform downstream operations. For example, if you have a thick cell wall, which in this case is not applicable, but for example, in case of microalgae, typically cell walls, if they are very thick, it's difficult to break them and extract protein out of it. So any such challenges which can come downstream can be addressed through strain selection. And of course, you need to, while you have to screen for all these different parameters, you have about 100,000 plus species from which you can select, and then specific strains within the species. So there's a lot of room to explore. Feedstock selection majorly plays into the cost of the end product, because there are only two incoming ingredients. You have the microorganism with which starts with a starter culture, and then you mostly have the raw material, which is then consumed and transformed by the microorganism, and the microorganism can grow on it or transform it in a way which is useful as an end product. So reducing the cost of feedstock is extremely critical to reduce the overall cost of the end product. It is also important that the feedstock is selected in such a way that for every kilogram of feedstock, it is efficiently converted into either whole biomass or the protein. Efficiency again refers to the fact that how much of your biomass that is produced is has how much content of it is the target compound. So if your target compound is a specific enzyme or protein, what is the efficiency of the feedstock to be able to provide that microorganism with that particular compound. So feedstock selection plays an important role, and we should keep in mind that feedstock is not just sugar, glucose, or beets or molasses, but feedstock also includes micro as well as macronutrients which can play a very critical role in the growth and productivity of the whole process. Then looking at selection, the next step, once you've identified, which strain to use, which feedstock to use, choosing the right production system is also critical. And here I've shown 3 production systems, and this definitely is not the exhaustive list. There are many different types of reactors which are available and can be easily found if you refer to a textbook on biotechnology. But I will touch upon the ones which have been used for some of the common applications. So first one is a stir tank reactor for yeast production. So typically this is a fed batch system, and it consists of an agitator system, heating system. It will have a system for with gaseous exchange. So basically, for yeast production, which is used for baking applications, the ratio between the concentration of sugar that goes into the feed and the oxygen in the air is extremely critical for its growth rate. So maintaining these conditions in a stir tank reactor are of most critical importance, and this is the kind of reactor used for yeast production typically. Then there is Lift Bioreactor, which is used for filamentous fungi production, and this here aeration is extremely important. And as you can see, this particular design is in such a way that the at the bottom you have the microbial starter culture, and then it rises with the bubbles that are injected into the system and gets circulated until you achieve a certain concentration of the biomass. And this is present in public knowledge that corn has been using airlift bioreactors for the production of corn products. Then the one that you see on the right hand side is an indoor solid state fermentation design. Typically, a mushroom cultivation unit will not be so complicated. It will A typical mushroom cultivation unit will have trays and a controlled humidity and temperature setting. But then, depending on the application and especially the kind of sophisticated growing conditions that we are looking at, which will be relevant for the smart protein sector, you need to be able to measure the oxygen levels, the carbon dioxide levels, uh, and control the local temperature, pressure, as well as humidity conditions very closely. So this is why this diagram looks very complicated and might be more relevant when it comes to smart protein applications. Once you've chosen a particular bioreactor system, there are the next step is being able to optimize the bioprocess. And this is where bioprocess design comes into picture. And you have several parameters that you can play around with. These could be biological, physical, physiochemical, and mechanical parameters. And these again are controlled by the choice of microbial strain you are using, the feedstock you are using, the design of the bioreactor itself, and the processing conditions in the bioreactor, which are dictated by temperature, pH, gas phase composition, rate of aeration, particle size, and the design of the agitator, which will define the amount of mixing and agitation. So you can play around with these parameters to optimize your reactor conditions for your production system. Last but not the least, you have the downstream operations, and this particular chart is relevant for applications which require extraction of a protein ingredient, or something like an enzyme, or vitamins, for example. This will not be relevant, for example, if you want to use the whole biomass, which can also go into alternative protein application. But let's say you want to extract protein and then extrude that protein to create a product, then this particular extraction process will be relevant. So typically after harvesting, you would need to do some kind of cell disruption, and after cell disruption, you need to separate the target molecule from the rest of the compounds, get the intracellular product, concentrate it, and then purify it. So depending on what level of purity you need, the concentration processes that can be used can differ. So anything from isoelectric precipitation to gel electrophoresis would depend on what is the target purity that is required. So you can see that there are two chains here. So the top one, which I mentioned, is when you are looking at extracting a product which is intracellular in nature, which will be the typical case for smart protein applications. The second one involves use of extracting components from the media itself. So you're not using the microorganisms, you are extracting stuff from the media, which may have enzymes or improved quality of substrate. So the second process is more relevant for a substrate, where again you have to do selective separation, concentrate, and then purify it. Now, coming to what would be the key differences between the liquid state and solid state fermentation value chain, so that you're able to visualize how the process happens. So let's take, for example, yeast or filamentous fungi, which require liquid state fermentation. So first step is obviously strain selection. I've explained that in detail. Then you can see in liquid state fermentation, you have a bioreactor, a typical stir tank reactor or airlift reactor, packed bed reactor, depending on what your goal is. And once you have, so this, in any case, this reactor will have majority of liquid component in it, and then there will be biomass, which will, the solids content will increase with time. After you have produced a certain level of microorganism in it, you need to ensure that the RNA is reduced. So typically filamentous fungi and yeast would have about 10% of RNA, which is not desirable. And to reduce it to 2%, you would need to heat it at a certain temperature to destroy the RNA. In this process, sometimes the protein also get destroyed, so you need to account for the reduction in the protein content. After RNA reduction, the next step is to be able to separate the liquid component from the solid matter, which has the microbial biomass, and then concentrate it subsequently, have higher percentage of solids, process it according to your needs. So this post-processing would involve any kind of downstream operations you need. And then from that, whatever ingredient is of your interest, you can develop a product from it. In case of solid state fermentation, your strain selection would involve taking up any of the mushroom species which are available, ensuring, like I mentioned in the strain selection process, ensure that it's safe to consume and make sure other parameters are being met. Then this will undergo solid state fermentation, and as you can see in the figure, there are trays in which you can like grow this, so these fruiting bodies, and depending on the complexity, like you may have a typical mushroom looking like product, or you may have like a big slab of mushroom biomass. I will show like one of the companies which is trying to do this. Now, in this case, in case of solid state fermentation, as you can imagine, there is no complexity in harvesting, because this is a continuous mass which you can hold in your hands. So in case the moisture content in the mushrooms is high, you can take it through a drying process, sterilize it. But if you want to use the whole biomass as it is, you can just directly make a product out of it, flavor it, marinate it, etc. However, there are people who have also tried to extract protein out of it. So they have once they have harvested the mushroom, they will the biomass would need to undergo all operations. So it needs to be pulverized or ground into small particles, and then the protein extraction processes, which could involve isolated point precipitation, chromatography, centrifugation, etc., can be carried out. So basically, protein isolate can be also prepared from mushrooms. Now, this was going to be a poll question, which I'm anyway going to read out. So the question was basically, which of these is a form of smart protein application of mushroom? And the options were protein isolate, whole cut meat analog, bacon strip, only A and B, all of the above, and none. So as you can imagine, with all these kind of questions, it's usually all of the above, and this is the case here as well. And I'll show you how that happens. So when we look at smart protein applications, I talked about protein extraction, right? So yeast has up to 50% protein typically, and mushrooms can have anywhere between 15 to 40% protein depending on the species. So you can basically extract protein out of these just like the way you would extract proteins from plants, and then use it in the extrusion process to create fungi-based meats, for example. The second way you can look at is creating meat analogues. So in case of filamentous fungi, as I said, so the way it works is that the filamentous fungi has this fibrous structure, and because it has this fibrous structure, this whole biomass, if you can compact it and bring adhesion to these to the different filaments, you can actually create mince meat analogues. So basically, this is what like most of the companies which are making filamentous fungi are trying to do. But there are also companies who have tried to structure these filamentous fungi in such a way that they can create whole cut meat muscle analogs as well. And this is one of the figures, if you remember, was in the case of meaty foods, and they are trying to make whole cut meat analogs. Then we have mushroom whole cut meats, and this is what I was referring to when I said that you can grow mushroom in such a way that instead of a small fruiting body, it is like a big continuous chunk of mushroom. And this is what Atlas Food, for example, has managed to do this. And this photo is actually from their website. The fourth application is basically yeast derived flavors. So yeast has an umami flavor associated with it, and this has traditionally been used to enhance the flavor of beef broths and other meat products, especially in the South Asia. But these yeast derived flavors can also play an important role in the cooking, marination, and flavoring process of meat analogs, and these could be plant-based meat. These can simply be used as flavoring agents. Then I had mentioned to you about how how fungi can be used to improve quality of substrate. So mycelia fermented proteins is something which is another application. You can improve the quality of plant-based proteins using fungi. Last but not the least, this is an amazing application of mycelium scaffold. So a spin-off company of Ecovative Design has recently, they have launched this product which uses mushroom mycelia to create scaffoldings which can then go for cultivated meat applications. And they have already been supplying this material to various labs to test out the functionality of these scaffolds. Having said this, all these 6 applications are not something which I have made up. All these actually exist in the market. So if you look at some of these examples, for whole cut meats, we have Atlas Food and Savory Wild, which are able to create these. And I would also add another company which is Meaty Food, which I mentioned before here. Then there's textured meat analogues, which involves which has been explored by many companies, and this includes CF Bio, Protein Brewery, Mycorrhiza, Mycovation, which is an Indian-based company as well. We have protein isolates, which are again coming from yeast as well as mushrooms. So Photortech is using mushrooms, oyster mushrooms, to extract proteins. We have More Foods, which is using protein isolates derived from yeast to create products. So they are into product development. They're not preparing these protein isolates, they're trying to use them. We have Next Farm and Yeep also into production of protein isolates, and Fumi Ingredients, which has used yeast again for creating egg white proteins. For flavor compounds, this is a fairly it's actually a market where big players are operating. So Angel Yeast, as many of you would know, is a leading player in the Asian markets, and Biospring La Safre from France. Both of these companies are actively looking at preparing flavor compounds which can go into smart protein applications. MedFood Lab is another company based out of Israel. This is a startup, and they're specifically looking at enhancing quality of plant-based meats using yeast. Then scaffolding materials, I mentioned a spin-off startup from Ecovative Design, which is Excel Scaffolds. They are trying to make these scaffolding materials for cultivated meat applications. Then last but not the least, fermented protein. I already mentioned Mycotechnology is improving the quality of rice and pea protein using shiitake mushroom mycelia. Now, this establishes the fact that one, fermentation as a technology is promising, and fungi is the most relevant microorganism from the perspective of smart protein sector. There are microorganisms like microalgae, fungi, bacteria, which can be used for various food applications, but so far fungi is the one which is leading applications in this sector, and you can see the diversity of applications that this microorganism has to offer. Now, having said this, what are the different opportunities we have in this space? How can we accelerate this sector? How can we leverage some of these for our work? So there are opportunities at each stage of the value chain. So be it strain selection and optimization. So this strain selection and optimization is so critical, as I mentioned before, and we need research institutes, we need labs which are able to do high throughput strain selection for people who are looking to do any downstream development process. So be it someone who wants to manufacture, be it someone who wants to do protein extraction, someone doing a strain selection at a high throughput is an essential need for the sector. And another important aspect here is that yeast is something which is very readily available in the market and has been available for a very long time. But what we don't know is that there are so many species or strains of yeast, not species, but strains of yeast which are being used both for brewing as well as baking applications. But we don't know which of these have superior functionalities for plant-based applications. So if it were possible to just have a repository of yeast strains and their functionalities in one place, that would immensely help the sector. So obviously open access research is important and like especially relevant for strain selection and optimization, but also for feedstock optimization, because India has a glut of sugar. And if we could use excessive side stream from agriculture, low cost substrates for production of yeast or filamentous fungi, this will immensely help in bringing down the cost of the overall process as well as the end product. And the next step, which is liquid state fermentation. In India, liquid state fermentation is something which people have been apprehensive about investing in, given the capacity and the scale and the volumes that are required for installation, and hence funding support is extremely critical for people and entrepreneurs to be able to venture into this sector. I would also say that for solid state fermentation, like India has a huge opportunity to venture into this, given small holders can explore the sector, and I'll talk about it later, not here. Then there are organoliptic properties which need to be determined. So many of these opportunities are very relevant for researchers in this group who are attending this session. So organoliptic properties is quite critical, because unless you know how good these fungal strains are when they go into plant-based meat or like fungus-based meat, for example, it's hard to do product development without this kind of a data. So determining how superior they are, how do they compare with plant-based proteins, will be extremely critical to accelerate product development in this space. The second last step, which is protein extraction techniques, is something which I think is a fairly mature technology, but has not been specifically used for fungal applications. It is mature from the perspective of biopharmaceutical industry, but for relevance in the food sector, this need to be again looked at and optimized for. And lastly, product development. Obviously, people who need to actually come up with products and bring these products to the market, so that there is building of consumer awareness as well as consumer acceptability, just because the access is increasing. And for all these, talent pool building is critical. We have a lot of biotechnological universities in India who are capable of working in this field, but they need to start actively exploring this and building skills, especially from food science come biotechnology perspective for the smart protein sector. Last but not the least, policy support is also critical, because as I mentioned, there is an investment component associated with liquid state fermentation. So easing funding opportunities, in easing their ability to establish this infrastructure will help. At the same time, also ensuring that there is ease in terms of labeling and getting these products on the shelf, getting the approvals would also be critical. So educating the policymakers and making them aware about the growing applications of fungal biomass will be critical for the development of this sector. What are the key opportunities in India? So these were the opportunities I shared in the last slide were more generic in nature, but in India there are two main big opportunities that are out there. One is leveraging solid state fermentation for protein production. So as I already mentioned, like it's solid state fermentation is simply mushroom cultivation in a way, but again it could be sophisticated if it could become sophisticated, because one, you need to select the right species and strains which taste like meat. So there are specific species of mushrooms which have a taste like meat. So for example, oyster mushrooms or chicken of the woods, there are several of these species. If you can see a list of them in our report, several of these species which have the flavors which one would look for in a meat. And not all of these mushrooms are actually grown in India. If we could, one, select these species, and second, optimize the process conditions, which is the most important part here, which requires scientific and research investment to be able to grow them in a fashion which is most productive and efficient. Given that, given the overall simplicity, overall lower investment compared to liquid state fermentation, this could be a promising option for smallholder cultivators in India. The second opportunity for liquid fermentation exists in terms of lowering the overall fermentation cost. So one is utilizing indigenous feedstock. So as I mentioned before, India is an agricultural country. We have food. There are side streams which come from agriculture, and there are side streams which come from food processing industry, and there is a scope to be able to utilize that. And hence bring down the cost. The second is bioprocess design innovations, and this is something which I believe is very unique to India, given the talent expertise that comes from the fact that we have so many biotechnologists trained in India, as well as the ones who are employed already in the pharmaceutical and biopharmaceutical industry, whose skills are actually transferable to this sector. So if we could design bioprocesses in such a way that it brings down the cost of production, that is something that can actually happen in India. And as I mentioned, right now we have the biopharmaceutical expertise already. We just need to leverage it for the smart protein sector. The last point I would like to touch upon is looking at decentralized fermentation as an option. Mostly fermentation is associated with big bioreactors of huge scales, but there is also opportunity to unlock capacities and capabilities of smallholder mushroom cultivators. So this goes back to solid state fermentation. We also have to, if we have to look at liquid state fermentation, we can probably think about fermentation processes that humans have used for many years for making food products at their home. So things like kimchi, soy sauce, tofu, tempeh, etc. have been made at home. Even kombucha for that matter. Many people buy the SCOBY starter culture and then use it to create kombucha at their home. If we could create such manuals which ensure that you can perform safe practices while using the microbial culture and grow them on your own, this provides an opportunity for small businesses to be able to operate at a smaller scale and still provide solutions to local communities, especially in resource constrained areas where there could be protein deficiency and people want to meet the nutritional requirements of the local community. I also want to highlight the fact that fungi especially have the advantage, especially yeast and filamentous fungi, that they have high protein content up to 50%. They have low fat, they are fiber rich, and can have complete essential amino acid profile to meet the nutritional requirements. So not only the alternative protein sector, but also the larger nutritional addressing the malnutrition concerns in a resource constrained area is something which can be addressed using fungal fermentation. Now coming to the last leg of this webinar, which is launching a strategic analysis of fungi report. So if you want to know more about, and if you were intrigued by this webinar, I would suggest you dive into GFI India's Technological Review of Fungi for Alternative Protein Applications. This particular report, the main focus has been the analysis of the state of technology for each application area. So the value chain and each application area has been discussed in detail. We have also taken 2 case studies. So one case study of baker's yeast, and one case study of corn, where we have explained how the process looks like, what are the reactor requirements, what are the feedstock requirements, etc., for you to be able to understand what are the process requirements. And we have also looked at the market development that have happened and the potential market developments that can happen in this space. Last but not the least, we have also done regulatory analysis and understood the policy landscape in India and identified some of the opportunities in this space. And also looked at the maturity of talent in India in terms of what courses, what universities are relevant for developing the expertise which would be relevant for for getting people working in the fungal fermentation for smart protein sector. Yeah, with this, I will just move on to my last slide, which is the opportunities dashboard. So in addition to the report, we also have an opportunities database, which is hosted on our website, which talks about the different interventions we have identified in India. And these interventions span from academic research to corporate to policy support, and relevant for different kind of stakeholders. We have identified the impact and the ease of implementation, and this has come out from our roundtable discussion that we had with key stakeholders in the Indian market, as well as interviews with academicians, industry players, startups, policy and investment firms, which are very closely working in the fungal protein space. With this, I would just open up the floor for more questions, and yeah, we can go ahead. Before that, I just mentioned that if you have any questions about the analysis, please reach out to scitech.india@gfi.org. I'll stop sharing, and I can take on questions now.
Thank you so much, Rama, for that wonderful session, despite of some of the hiccups we had on the operational side. And thank you all the attendees for patiently, having your microphones and videos on mute and, letting us conduct a successful webinar. So thank you. And I really hope that you all found it quite useful and intriguing. I've shared a few links on the screen, and I think you're already going through them. But once you kind of land on our website, I can quickly show you how you can join the GF Ideas India community, which is one of the largest communities of smart protein stakeholders in our country, where we are essentially having hundreds of entrepreneurs, investors, corporate stakeholders, researchers, scientists, students in this community. And we also do quite a lot of initiatives for different stakeholder groups throughout the year. So stay tuned and do sign up for the GF Ideas India community. This is our website. You can take a look at a lot of our key resources directly in the resource section. I've put in the link in the chat box. You can also visit some of our other stuff to get in touch with us, know about our team, understand about our mission, and some of the resources and perhaps open access kind of tools that we have built for you. This is the page when you click on resources, and when you essentially click on GF Ideas India community, you are given a link to essentially join us and read through about the community, kind of understand all the resources that you would be able to access in the community, etc., etc. So, all our past recordings for the 19 episodes and the 20th episode, which is today in the GF Ideas India webinar series, would be all available on a YouTube handle. And, all of the previous recordings are already here from our previous summits, from our previous webinars, and a lot of our, India Smart Protein Innovation Challenge webinars as well. So please feel free to take a look at all of that. I'll stop sharing. And with this, I think, Rama, we can quickly take a couple of questions that we had received from the Zoom registrations today, and then we can take a couple of live questions as well. And we'll extend this webinar by 5 minutes, and we'll close down at 6:20. So thank you for your patience, all the attendees. So the first question, Rama, is protein quality that is developed through fungi. And when we talk about plant-based sources and other alternative sources of making protein, how does it compare? And how do we reduce and minimize the risk of toxic metabolites developed in the process of fermentation, which kind of limits the current use case of fungi as an alternative source of protein in many ways, right?
Yeah. Yeah, thanks for the question, Shardul. So in terms of the protein quality, as I mentioned, it's one thing I'd like to mention before talking about protein is that when you look at the whole biomass, it's actually better to use the whole biomass, because you're not just getting protein, you are getting low fiber, you are getting no fats, you're basically getting all the vitamins and minerals which are essential for us. And if you're looking specifically at protein, I think there has been good literature in terms of what is the PDCAS values, which basically determines the protein digestibility of mycoproteins. And this in the scientific literature is around 0.996, which is close to the PDCAS values for chicken and milk. And the mycoprotein typically contains all essential amino acids, making it a complete protein source. And the bioavailability of these can be have been demonstrated to be equivalent of milk protein, and sometimes even significantly better than wheat-based protein and soy-based protein. So definitely, if again, like I would caveat this by saying that it depends on the species that you're looking at. So this mycoprotein, I believe, is a fusarium strain, but depending on the strain, you need to do these studies. But early data does suggest, like the existing data does suggest for these species, it has superiority on some of the aspects for sure. And in terms of mycoproteins, mycotoxins, so there are two things of interest. One is that you need to ensure while screening of the strains that these don't produce mycotoxins. In case you are using these strains which produce mycotoxins, probably one way to mitigate that is by using growth conditions or media which prevents that production pathway for mycoproteins. So these are two factors that you need to keep in mind. And I think from a food safety perspective, having mycotoxins will not be acceptable. So one needs to ensure that that doesn't happen.
Got it. On that note, I have a follow-up question from the audience as well about studies into the potential downsides of, chitin allergies linked to high levels of mushroom fungi consumption. Have you come across any of those, or is that something you can look up and perhaps share later?
So far I haven't come across this, but I'm sure that like depending, as I said, that fungi has so many species. Depending on the species that you're using, the allergen potential would vary. So you need to if you're using for food applications, you need to screen them for allergenicity for sure. So again, this is not generalizable. It depends on the species you use. And of course, like there might be studies. I'm not aware of that as of at the moment.
Makes sense. So the common theme that was coming out of the many questions was also difference between algal proteins and fungal proteins, especially when we talk about the applications in the alternative protein sector. So can you throw some light about that?
Yes. So for those of you who are not aware of this, GFI India had launched the microalgae and seaweed strategic analysis in the month of January. And Shardul can probably drop in the link to the webpage, and it also has the opportunities database as well as the detailed report which you can look at. The basic difference I would say is that, like again, like fungi has so many applications, but in case of microalgae, you are restricted by the fact that you need to extract protein out of it. In many cases, at least until as far as till now, what has been reported, microalgae requires you to separate other components from the protein, because you'll have the color and then the oils and lipids, which you don't want in the end product. So for microalgae, it's quite straightforward. You cultivate the microalgae, which is typically like the productivity rate, the productivity as well as the production rates are lower compared to fungi, but depending on the process that you use. But basically, you'd need to go through the entire process from production to extraction for sure. But in case of fungi, if you use, for example, filamentous fungi, you don't need to. You basically don't need to do the extraction process to start with. If you are using, and also like in terms of cell wall, like microalgae is known to have like a tough cell wall, and hence it's difficult to break, energy intensive to extract protein. This will not be the case in case of yeast, for example. It's simple. You can use standard thermomechanical processes to break open the yeast cell and obtain protein out of it. And in terms of protein properties, this is something which has, I would say, is not very well reported. We just know in terms of like through the value chain how the processes differ, but not the protein qualities.
Makes sense. Thank you, Rama, for sharing your insights. I think one of the last questions which kind of coming up is on the regulatory side. The theme emerging is about regulatory status for all of the ingredients that are, you mentioned today from fungi, all the proteins that we are kind of producing from mycelium fermentation. So could you talk about their regulatory status?
So as of now, if you could find more detailed information in our report, but there are very few generic mushroom species that are approved as agricultural products that can be used under different food applications right now. So it's mostly the agaricus species which has been approved by the government. For other species and formulations, especially like mycoprotein and yeast-based protein, you'll have to apply under Nobel's food framework as non-specified food for food products and food ingredients. Yeah, so basically it's something which requires more proactive awareness to policymakers to make this more mainstream.
Makes sense. Yeah. Thank you so much for sharing those insights. Unfortunately, we are over time. But as Rama mentioned, please feel free to get in touch with the team for any specific information that you're kind of seeking information on. Other than this, just checking the link that Joanna has shared. Let me check if it's working. Hi. So I think the link's working. There's a small, box where you can fill in your details, and then essentially it will send the report to your, inbox. If it's not doing that in the next 15, 20 minutes, we'll get it checked and essentially fix it if it's broken. But I think you should have the report
Shardul,
link
I
in your
think the problem is this had happened before as well. So you need to fill in all your details, even the ones which are not optional. If you are filling only your email ID and first last name, it will only show. It will not show the download link. You need to fill in your company, job title, and industry details as well. If
Got
you
it.
fill that, you should be able to access.
Yeah, please fill all the details to access the copy that comes into your inbox. If you leave a field open, it won't come into your inbox. And feel free to try the link again tomorrow. Sometimes the cookies, etc., are also a problem. And I think it should be live, and we'll fix it at the back end. Thank you so much for joining us, everyone, today. And we hope that you're able to, join our community if you're a specific stakeholder in the smart protein sector or the global alternative protein sector. If you have skill sets, business background, or potential interest in starting something in this sector, we'd love to have you in the GFI Ideas India community so that we can share tons of other free open access resources with you, which are essentially relevant to get you started on your own individual paths, whether as an entrepreneur, as a student choosing a career in this field, whether it's about fermentation and fungi or algae or cultivated meat, eggs and dairy, or plant-based meat, eggs and dairy, etc. Thank you so much.